Good morning, everyone. Thank you for joining the webinar this morning. My name is Liz. I'm the co-speaker and the moderator for today's webinar. I'm here with Melissa Smith, who is our guest speaker. A few housekeeping items before we get started. This webinar is going to play recorded talks. My intro talk first, followed by Melissa's. We will have a live Q&A at the end. During any time of this webinar, you can use the questions box to ask questions that we will answer at the live Q&A at the end. So if you're in your GoToWebinar, if you take a look at the upper right corner, there is a little box with the question sign. You can ask questions in the questions box. However, we will only answer these questions at the end of the recorded talks. Second, you can also check out the resources tab, or actually the materials tab, which is next to the questions box for collateral related to today's webinar. This webinar is recorded and you will receive a recording in your inbox a few weeks later. Finally, we'd appreciate if you can fill out the survey at the end of this webinar. Thank you, and let's get the webinar started.
Good morning, everyone. Thank you for joining the webinar today. My name is Liz, and I'm an Associate Director in Product Marketing at PacBio. Before we get to our guest speaker today, I'd like to give you an overview of how one can use multi-omic discoveries in cancer research with PacBio long-read sequencing. PacBio HiFi sequencing enables comprehensive multi-omic solution for cancer research. This includes the ability on the genome side to detect germline and somatic mutations, where the accuracy of the reads enable precise small and large variant detection, while the long read lengths enable phasing of these variants. With native DNA sequencing, we can also see methylation patterns. Later, we'll show that we can also obtain chromatin accessibility information using a technique called Fiber-Seq. Applying long reads for the transcriptome, we can see full-length transcript isoforms, fusions, and mutations on the transcript that can enable allele-specific isoform detection, or in the case of single-cell, clonal tracing. Our end-to-end DNA workflow solution starts with high molecular weight DNA extraction using PacBio's nanobind kits, followed by library preparation with sequencing on our long-read sequencers. An additional step can be taken before DNA library preparation using a technique called Fiber-Seq that allows the ability to examine chromatin accessibility without additional sequencing. The way Fiber-Seq works is to take cells and use a methyltransferase to artificially label the accessible chromatin regions with dense 6-methyl-A marks. Those are these little purple asterisks. Then, DNA is extracted and made into a library for sequencing. The readout for the 6-MA marks identify regions of open chromatin. So in a single assay, you get the genetic variants, the 5-MC methylation status, chromatone accessibility, which lets you look for new types of biomarkers related to gene regulation. We also have a software workflow. This is a WIDO workflow that gives you the input file and output small nucleotide variants, copy numbers variations, structural variations, differential methylation, homologous recombination deficiency. Next, I want to talk about some of the recent improvements in our product offerings. Last year, PacBio rolled out a new sequencing chemistry called SPARC that enables 4 times less DNA input for PCR-free libraries and increases output by 33%. So you can get a 40-fold human genome per SMRT cell. This also means that a lot of samples that didn't have enough material before are all now accessible with HiFi. We also released an amplified protocol that enables HiFi sequencing with as little as 1 nanogram of DNA input through a whole genome amplification approach. This enables the use of challenging sample types with HiFi, including FFPE. For the first time, it is now possible to sequence FFPE samples with HiFi. While we are not getting long 15 kb reads as we would with regular high molecular weight DNA as you would with a fresh frozen sample, this is still better than using short-read technologies. In this use case, we demonstrate that on FFPE samples with Amplify, we get a mean read length of almost 3 kb with a tail out and out to 8 to 10 kilobases. The quality of the reads is really high because these short fragments mean many passes go through the circular SMART-bound molecule. Because these fragments are also shorter, we will get slightly lower HiFi yield than regular large DNA fragment libraries. Moving on to RNA sequencing, HiFi provides unique capability to sequence full-length isoforms both at the bulk and the single-cell level. When using short reads in bulk transcriptomes, transcripts have to be computationally assembled, which becomes increasingly unreliable for genes that have complex alternative splicing patterns or novel isoforms. Meanwhile, in single-cell, short reads simply do not capture isoform information, but only the gene information. What can single-cell isoform sequencing with HiFi bring that short reads or other technologies cannot? In one case, researchers applied single-cell IsoSeq to ovarian cancer samples and found patient-specific, tumor-specific fusion isoform. Using matching short reads, the short read analysis misclassified the fusion reads to one of the component genes, TESPA1, because it can only capture a small portion of the transcript. In contrast, long-read sequencing accurately identified the fusion transcript in the cancer cell. In another case, researchers used the mutations found in single-cell transcripts for clonal tracing on chronic lymphocytic leukemia patients that had developed drug resistance. Using long reads, 32.5% of the reads covered almost all of the canonical transcript length, allowing for a high degree of genotyping. In contrast, with short reads, only 0.1% of the reads have good coverage of the transcripts. Using the long read data, improved the subclone structure on one of the patients previously based on short read whole exome sequencing data. This is because HiFi has the ability to phase through multiple variants on a single transcript. Finally, single-cell IsoSeq shows promise for neoantigen discovery. Researchers applied this to colorectal cancer samples and discovered novel isoforms, RNA editing events, isoforms associated with cell type differentiation. But the most promising find was identifying neoepitopes that showed promise as vaccine candidates. To sum up, we're really excited about what HiFi has to offer in getting a complete and multi-omic understanding of cancer. Thank you.
Hi, I'm Melissa Smith. I am an associate professor here at the University of Louisville in the Department of Biochemistry and the CEO of our new startup, Clareo Biosciences. I'm super excited to share our results today on precision immunogenomic profiling in cancer, and really want to thank PacBio for the opportunity to give this talk today. So I want to start the talk by telling you a story of two grandfathers. So one grandfather is my grandfather's age, diagnosed with metastatic melanoma in the late 1990s, let's say 1997. At that time, there were no real options for this disease, and advanced melanoma was given a diagnosis of sort of 6 months left to live. And so that was what you had, and that's unfortunate. Now, a grandfather today who gets diagnosed with advanced cancer, maybe advanced lung cancer, has the opportunity for immunotherapy. These are drugs that activate your immune system to fight the cancer itself. And in this case, this grandfather does take the immunotherapy, does respond well. His tumor goes into remission. He's playing with his grandchildren, and 2 months later develops an inflammatory kidney disease that ends up being fatal. And this is actually not that unusual of a circumstance. 64% of patients that receive immunotherapies benefit from them, and it has a positive effect on the tumor. But unfortunately, 60% of those patients also develop immune-related adverse events, or IRAEs. These events can range from very nondescript, maybe some small sort of digestive issues, all the way to more advanced stage, potentially lethal outcomes. Now, what we do know is that the adaptive immune system, particularly the T cells and the B cells, are responsible both for response to therapy as well as these immune-related adverse events. And our team has done a tremendous amount of work that I'm going to show you today, defining the variation in these genes across populations. And we have linked this variation to response to different therapies, particularly vaccines. And so what if we could use that information to inform future therapies to both enhance responsiveness or at least predict responsiveness, so you know the drug you're going to take is going to have an effect, and reduce the chance of off-target effects. So why is this new? Why has no one done this before? Well, for many, many years, we were doing disease association studies using array-based technology, or GWAS. And what I'm showing you here is a density map of the oligoprobes across the entire human genome. And when you look specifically at the variants that are represented in this array in immunogenomic loci, or those genes that encode our immune system genes, you see that there are genes that we know are really associated with disease, like HLA. Some consider this the most polymorphic region of the human genome. Even less well-characterized regions, like the KIR receptors that are involved in NK cell action, are reasonably well covered with hundreds of variants reflected. But the adaptive immune receptors, both the immunoglobulin that encode antibodies and the T cell receptors that encode T cell responses, are not well covered at all. In fact, here's an example of the density of SNPs in IgH that was on this immunoarray. There's only 5 SNPs in over a megabase of content. I can tell you that this does not reflect the variation in this locus. And this meant that those disease association studies based on this data basically ignored these regions. And so we don't really understand, although it is intuitive to think that our immune system genetics would be involved in the response, how that is done, what that mechanism is really unclear. So let's show you why this was an effect and what the trickle-down impact of that has been. So for those of you that have taken immunology in the past, I don't want to spur any post-traumatic stress by putting this up, but understanding the structure and the mechanism of VDJ recombination and its role in encoding the receptors of both antibodies and T cell receptors is critically important for us to move forward and talk about how HiFi data has really enabled our ability to see a complete picture here today. So this is a simplified schematic. And to remind everyone that hasn't thought about this since a college class, this locus undergoes somatic recombination. So literally cutting chunks of DNA out between the V, D, and J cassettes that are then ligated together with non-templated nucleotides in between to create that VDJ cassette, that variable region that recognizes antigen, either in the context of an antibody or a T cell receptor. Once that cassette is done, it's then spliced into constant region cassettes to encode the constant region of those receptors that then perpetuates downstream functions. Now, as I mentioned, this is an image out of Janeway's immunology. This is very oversimplified. If you actually look at a map of what this looks like in the genome, this is what you see. Rather than simple cassettes aligned together, it's quite a massive mess of variable genes, diversity genes, joining genes, and constant genes that are all interspersed with those red blocks that are pseudogenes. So these were variable genes that evolved and acquired non-functional mutations, rendering them non-functional in the repertoire. The big issue to point out here when it comes to sequencing technology is that those green cassettes are about 300 base pairs, and then they ligate to the D and J cassettes to make that entirety of the variable region, and they're highly repetitive. 50% of this locus is within one segmental duplication, and each of these can be separated sometimes by only a few coding residues. That means that using a short read technology that barely spans, or maybe even doesn't span, the entirety of that gene cassette, and then trying to stitch them together in an assembly of this entirety of this locus is almost impossible with short reads. People sequence through it, but achieving an accurate assembly is incredibly difficult, if not impossible. So starting almost 11 years ago, my colleague Corey Watson here at U of L and I started to work at figuring out if we could use long reads to resolve these haplotypes, define these genes, and understand the structure of these loci, and then examine how the variation we saw at the DNA level impacted the antibodies and the T cell receptors that we saw at the RNA level. So we've developed 2 novel assays that I'm going to present to you today, both a genotyping assay outlined in the red box with the genotype schematic on the top, and a near full-length repertoire profiling assay surrounded by the blue box with the little antibodies above it. These symbols I just put up there because I'll keep them in the corner of the slides so that the data that you see moving forward, you can tell immediately whether it was derived from DNA data or RNA data. So what I want to start with you is to really prove to you that we're not all the same. So again, when you think back to that Immunology 101 class, we're often taught that we all have the same antibody genes and we just mix and match them differently. And that's why maybe your friend in the office responds differently to the flu vaccine than you do. What I'm showing you here is a summary figure. We profiled the immunoglobulin loci or antibody genes in 154 different individuals across 5 major global subpopulations. And what is shown here is the extent of variation within this cohort. So across the top of this slide, you can see the schematic of the genes, the VDJ genes. So here we're reading actually from right to left because this is on the telomeric end of chromosome 14. So the V genes moving towards the centromere, the D genes, and then the J genes. On the next step down, you see a schematic of the structural variation that was seen in this locus. Particularly deletions are shown by these red dotted lines and insertions shown by these orange triangles. Now, I want to emphasize that each of these structural variation patterns were seen in at least 5 out of the 154 individuals in our cohort. So these don't represent super rare variants that we'll never see again. These are relatively common, about 5% frequency. And these include massive deletions that remove multiple genes from these people's DNA from the get-go. That's the foundation. That's their starting line from which they can mount an immune response. So if, for example, you were to design a vaccine to target this V320 gene, these groups of people that contain this large deletion would have really no chance to respond as designed. Similarly, if you were to design a vaccine that tried to stimulate particularly this V323D gene, that's only going to be inserted in certain populations. So the presence of or an absence of genes is something that is underappreciated, especially when it comes to therapies that specifically want to leverage your immune system to have their therapeutic effect. Now, these genes are not only differing in their presence or absence, they're also differing in their flavor, for example. So there's this allelic variation where the gene could be present, but a different form of that gene can be there. And this difference in form can be just one coding difference. But there are particular genes that are enriched in multiple alleles. And in 154 individuals, we identified 127 novel coding alleles. Again, this is just to drive home that we really don't know the extent of variation out there, and that this may be critically important for understanding differential responses across people. Now, this is immunoglobulin. I've trained as an antibody person to start with, but this is a feature that extends across the adaptive immune system. This is not just an antibody thing. This is also a T cell thing. So in a much smaller study, we studied 9 genomes from individuals that had been previously characterized in the 1,000 Genomes Project. So these were samples that had been characterized by multiple sequencing technologies. And even in that context, these loci had not been well annotated and well characterized because of that struggle of assembling short reads in this region. We use long reads to capture and resolve these haplotypes. And what I'm showing you here is that the genes here are aligned across the y-axis. These are the alpha, beta, and delta genes of the T cell receptor. Gamma isn't represented here. But even in these only 9 individuals across 3 major global populations, we identified almost 80 new alleles. So any box that is red with a black dot, that represents a novel allele that hasn't been seen in the database before. And then any block that is blue with a triangle represents a deletion. And you can see specifically here, there are population-specific deletion effects, as well as insertion of novel alleles in particular populations. So we're currently working to expand this to the sort of largest T cell genotype cohort to date. We've got hundreds of individuals we're currently characterizing. But given that immunotherapies in particular target T cell responses, this level of variation, although we were expecting to see that it was variable, this was overwhelmingly variable. It was exciting in the potential of what that could mean for therapeutic intervention, but also a little bit daunting in the amount of variation that we saw. So we were motivated initially to understand the role of T cell variation at the DNA level with the response to immunotherapy by this paper that came out in 2023. So this is a PCA that was taken from the Stephen et al. Paper in the Journal of Immunotherapy. This was a paper where the group led by Dr. Timothy Looney did TCR beta RNA sequencing. So they sequenced the TCR beta chains that were circulating in the repertoire, and then they inferred the germline content or the genotype from that RNA data. So based on the genes they saw in the repertoire, they could say, okay, those genes are also in the DNA. And then they associated which genes these individuals were carrying with the presence and severity of immune-related adverse events, or those IRAEs, like I discussed earlier. Now, in this case, there are no healthy donors. This is just a variable of severity. And what they did identify was that there were a set of genotypes that appeared to be protective, that if you carried those genotypes together, you did not have any immune-related adverse events, which of course is the ideal scenario from a clinical standpoint. So we were really curious, okay, this TR beta data is really convincing. What if we added the additional loci that we're capable of characterizing, and in addition, go from the RNA data to DNA sequencing, where not only do you see the coding variation and you're not inferring genotypes, but you're actually resolving the DNA directly. So we at ClareoBio, and we're going to talk about sort of ClareoBio as a specific entity in a little bit, we did this kind of profiling, a different cohort. And in this case, we had 55 individuals that developed immune-related adverse events in the context of immunotherapy, and 30 individuals that received immunotherapy and did not develop adverse events. And when we looked at their genetics at the three major Ig loci. So here you have the heavy chain, that's those VDJ genes I showed you a minute ago, and then the constant region genes, but these are both IgH, as well as all four TCR receptor loci. So if you remember, again, the Looney paper the Stephen et al. Paper I showed you on the left, these are the variants that we found sort of above the threshold for significance with correction from multiple variable analysis. We do see TR beta signals that are associated with the development of effects. So that's nice. It validates the previous study. But if you look across the genes, we see an incredible number of variants across all of the immune loci, or almost all of the immune loci we look at. Now, from our perspective, the more variants you can identify, the more potentially sensitive a biomarker discovery assay can be. So we were really excited about these results. Not only did they verify an older observation, but we think we have additional information to overall enhance some type of predictive algorithm that could be used for biomarker discovery to predict the likelihood of developing immune-related adverse events. But then we really wanted to sort of pull these data and unwrap them and see like how much content and how much explanation can we get out of just, this variant analysis. So we have done work in the past and published papers, as well as continuing in the laboratory here at U of L, looking at the impact of these variants on expression in the repertoire. And this is termed eQTL, or expression quantitative trait loci analyses. So this is just a cartoon of how it works, and I think it's important to interpret the data I'm about to show. So obviously, in this first example, if you have a gene and it's just not present in your DNA, then there is no way you will express it in your repertoire. And so if you look at these blue box plots on the right, the person who has the deletion as a homozygous on both chromosomes are not going to express that gene because it's just not there. And if you have a heterozygous insertion deletion, then you'll get sort of mid-level expression. And the hope would be that if you have two insertions on both chromosomes, you have the highest level of expression. Now, this doesn't just affect the presence or absence of genes. This also can be affected about SNPs that are upstream or downstream of coding exons. I think it's become abundantly apparent in the last few years that most of the regulatory SNPs in our genome are actually not in coding exons. They're upstream or downstream of genes, and this is no different in the Ig loci. And so in this case, you may have a SNP that's upstream or downstream of an exon or a V gene that inhibits the ability maybe of this gene to be recombined. Maybe it interferes with the RAG recombinase signal. Regardless, if you were homozygous for that AA variant, you express very little of that antibody or T cell receptor in your repertoire. If you're heterozygous, maybe you get that middle level. And if you're homozygous for the productive variant, you have the highest expression in your repertoire. So we wanted to look at those variants we found in our IRAE cohort and see if we could impute or infer any function from those variants. So we're going to zoom in here on just the IGL. So this was the box or the stack on the Manhattan plot that had the most variants above significance. We're going to spread this out across the loci and try to understand whether these variants have any type of functional association. So in particular, we're going to zoom in on this variant, so highly significant association with the presence of immune-related adverse events. We also saw that it was enriched in our cohort compared to the control samples that we had, the immunotherapy treated but no adverse events cohort. And then we wanted to look at what that was associated with when it came to expression of specific genes in the T cell repertoire. And what we saw is that the people who had this variant, the IRAE prone individuals, were more likely to impact the expression of this IGL V218 allele star O2. And really what I want to show you here is that the difference between these alleles can be incredibly minor. So the star O1 and star O2 alleles, they only differ by that leucine to serine at the beginning of the CDR3 region. So the CDR3 you may have heard of, it's the complementarity determining region. It is the most hypervariable region of these receptors, and it tends to bind with the antigen. So not surprising that that's where we would see the variation lying, but that it is really important that it can be just one residue change that changes the dynamic and the function of these receptors. So if you have this variant, you have a more likely expression of this star O2 allele at this gene. And this is shown here. So in the alt or that leucine, the variant that is not being there, you have a lower expression of this gene. And this was actually a different cohort. This is in our IRE cohort. This is a healthy donor cohort. So this is assessing the impact of this germline variant on sort of just general receptor frequency in T cell receptor dynamics. But this variant is really associated with increasing the expression of that IGL V218 star O2. And I think that's critically important to consider that suggests perhaps that this allele is associated with that effect. Obviously, there would need to be much more investigation to determine function and impact, but it does show that the variants we identify just with these association studies can start suggesting functional hypotheses that then we follow up on. Now, because at the root of what we do is population genetics and understanding how different people vary at these loci and how that impacts their susceptibility to disease, we took this variant and we looked at its distribution across global subpopulations. So in those 170 individuals, we had representation from African cohorts, East Asian cohorts, European cohorts, and Native Indigenous American cohorts. That ref allele that showed the higher expression of V218 star O2, that's represented there in pink. And you do see an enrichment of that allele in European specific populations. Again, I'm not drawing any biological conclusions, but it does suggest the hypotheses that if that variant and that gene expression is associated with disease pathogenesis, maybe this would differ across populations based on that genetic frequency. So in sort of wrapping up this very preliminary but exciting data, I want to draw attention, again, the conclusion is that variant is associated with the expression of that IGL V218 star O2 allele. Now, importantly, that SNP is intergenic. It's not in a coding exon, and it would not have been visible to inference-based methods. This is really the power of directly sequencing the DNA, and it's the power of HiFi reads. The long HiFi reads are what enables us to correctly assemble these loci because they're longer than those repeat elements. They really can push through them and assemble them correctly, and that allows us to map not only coding, but non-coding variants. We believe that the combination of variant identification across immune receptor loci is going to be really powerful to identify specific sensitive signatures to disease prediction. And we think that the data I'm showing you here is sort of our first glimpse of that. And we've developed these technologies to provide comprehensive pan-immunoreceptor genotype associations. Now, all of this is in the literature. Not this data, but these methods are in the literature, and those papers are cited here in these slides. So please feel free to dig in and look into it, and I'd love to hear what you guys think. So I'm going to pivot now and talk about immune receptors and their repertoires at the RNA level. So we're going to step back again and think about the structure of these molecules, because that's really important to understand why HiFi has been so powerful in us exploring them. So this is a schematic of an antibody transcript. The VDJ cassette, again, stuck together, glued together with those non-templated nucleotides. And that intersection between VD and J creates that CDR3 or hypervariable region that interacts with antigen. And then there's the constant region that are encoded by CH exons. Almost all of the IGs have 3 exons. IgM has 4 exons. And it's sequencing signatures within these constant regions that determine the isotype of the antibody. So whether it's an IgM, IgD, IgG, IgA, or IgE, as well as the subisotypes specifically within the IgG and IgA populations. Additionally, this constant region creates the stem of the molecule itself that interacts with Fc receptors, that interacts with complement in order to propagate effector function. And even though it's called constant, antibodies have different functionalities, suggesting that there must be some type of signature in this region that directs that function. We think that signature is genetic, and so that's what we set out to prove. So one of the reasons that the constant region hasn't really been well explored is technological. So currently in the market, there are three really big methods for looking at antibody repertoires and describing them in the literature. And all of them are very focused on the variable region. This is incredibly obvious to me. This is the region that interacts with the antigen. And so it makes sense that we'd want to understand that as a biomarker of specific infection or disease. So in some cases, like the assay that Adaptive Biotechnology offers, there's a profiling just of that CDR3 region. This is the hypervariable region. You can think of it as a fingerprint of that antibody. It's really short, very, relatively straightforward to characterize. Then there are methods that want to capture the entirety of the variable region that either use multiplex primer sets, which are very powerful, but require you to know what you're looking for. Given that we're constantly identifying novel alleles, I don't want to miss anything that I don't know. And so we tend in our laboratory, at least before the development of the method I'm going to show you, to use the rapid amplification of cDNA ends methods. This uses a primer in that constant region. It runs through the variable region, adds a template switch oligo, and amplifies back so that you can grab variable regions that may be unknown and discover novel alleles. Now, all of these you can notice have gray in that constant region structure. None of them characterize the constant region with any type of comprehensive nature. And that is because of the restriction of short read sequencing. So I have a MiSeq pictured here, an older MiSeq. The 2x300 kit, of course, is now available on multiple platforms from Illumina, but really that's the length restriction, right? 600 base pairs, probably about 550 if you want to make sure you can assemble them with some type of overlap in those paired reads. And this cassette ranges from 5 to over 600 base pairs in length. And so in the paper that I'm going to describe here, the results from, we actually saw that not only does our method capture the entirety of the molecule, and this method is FLAIR-seq, so we actually just move that primer down to the end of the molecule and capture the entirety of the transcript. But even some of the short read methods don't actually capture the entirety of the variable region if somatic hypermutation actually extends that region larger than the read capacity of the short read instrument. So let's talk more about FLAIR. So full-length adaptive immune receptor repertoire sequencing. As I mentioned, we use a rapid amplification of cDNA methods. We have a primer that is all the way down at the 3' end of the molecule to grab near full-length transcripts. Now, everyone thinks of PacBio as a long-read technology, and it certainly is. But what I think of it as more is a flexible length technology. So these transcripts are 1.5 to 2 kb on average, and you can sequence anything from 500 base pairs to 30 kb on the PacBio. And what that allowed us to do is not have to worry about whether the length was going to be a restricting factor. We could put the entire transcript on in a targeted fashion and ensure that we got adequate coverage no matter how that length was flexing due to ongoing somatic hypermutation. So initially, we sequenced these as single chains, these 1.5 kb inserts into SmartBell Prep. But what has been amazing to us over the last year or so has been the advances that were allowed by the Kinnex and Spark protocols. So Kinnex has allowed us to concatenate multiple transcripts and take advantage of that length space. So instead of sequencing 1.5 kb per reaction, we now sequence 8 of them glued together, essentially, for a 12 kb molecule that actually represents 8 transcripts tiled together. And that's allowed us to increase our depth 8-fold. In either way, you have an incredibly accurate data set, especially even in the concatenated molecule. We're still seeing our reads at above Q40 or 99.99% accurate. That's an order of magnitude over other technologies, and is critically important for these types of questions. That mutational pattern that happens during the evolutionary arms race between the host and the pathogen introduces point mutations that change the function of the antibody. If you see sequences and you can't be sure whether that variant is biological or artifact, it really hampers your ability to interpret your data correctly. So that accuracy has been essential for us. Now, I want to show you what these data look like at the actual sort of read level, just because it's been really powerful to me to be able to see how the entirety of the molecule linked together can provide additional power. So the variable region, of course, on the right of this molecule, again, we're reading from the telomeric end of the chromosome, is the VDJ. Yes, it's variable. Every color here represents an A, C, T, or G, and the mishmash of colors shows how crazy variable it is. But the constant region is not entirely static. There are SNPs across the constant region, and the gray are the exons here. This is an IGV alignment. The blue are the introns. And you can see the presence of SNPs that differ between the maternal and paternal allele, allowing you to get allele level resolution at the transcript level. Now, that's a really nice technological trick, but let me show you why you should care biologically. This is the current resolution you have with the kits that are on the market that only do the sort of race approach that grab just that chunk of the constant region that gives you isotype resolution, right? You can look at the unique clones that are present, within an isotype specific compartment. So here are your unique VDJs with an IgM and your unique VDJs with an IgG. Using FLAIR-seq and getting that enhanced allele level resolution, we can now break down which of those clones are in which compartment. So for IgM, which clones are associated with the maternal versus paternal allele, and for IgG, which are associated with which isotype, and then within isotypes, maternal versus paternal allele. We can visualize that with all kinds of fancy graphics. This is one of our favorites, this sort of Circos plot. So let me take a second to walk through this because I think it's critically important. On the top of this Circos plot are the different alleles that we're seeing in our data. So this pink bar here, these are the IgG1 maternal allele, and then those are linked on the bottom chord with the variable genes that are being used within that compartment. And what is really powerful here about this technology, the so what of the technical trick, is that if you were specifically interested in an antibody clone in the IgG3 compartment, if you look at this bar graph here, IgG1 and IgG2 really dominate about 90% of the repertoire. And so if you're really interested in an IgG3 molecule, you're fishing through a lot of noise. You're associating clonal expansion in IgG3 with Western blots of the constant region and allotyping, and trying to extrapolate an indirect association to try to pinpoint that IgG3 specific antibody. With this method, you can analyze it and say, no, all the noise is gone. Here are my two IgG3 alleles, and these are the V genes they're using. And that allows you to really hone your investigation to specific antibodies within that compartment you know you need. More than that, we can actually look at antibody evolution in semi-real time. So we demonstrated this in our paper by using samples taken from an individual who had was hospitalized for COVID, sampled over a period of days, and then did FLAIR-seq and understood how the antibody evolved. And what we can see is we can identify one of these single clones and then say, okay, is it an IgM, which is normally associated with a naive B cell that has not undergone class switch recombination, maybe isn't mature yet. And then over time, we can watch how that clone moves between the IgM compartment into the IgG compartment, suggesting that it underwent engagement in the germinal center, affinity maturation, somatic hypermutation, and now class switch recombination to get into that functional state it needs to be to have its antiviral effect. So really powerful technology to look at antibody evolution and really hone in on your specific clones of interest. Now, we wanted to demonstrate the effectiveness and the new value that we could add in the context of disease. And so we chose acetylcholine receptor associated myasthenia gravis. This is mediated by IgG1 and IgG3 antibodies. So there's been a lot of groundwork that's been done that really would benefit from that sub-isotype level resolution that I just described. We know that the mechanisms of action in this disease are threefold. So in this case, there are autoantibodies that target the neuromuscular junction, specifically the acetylcholine receptor, either through complemented mediated destruction, internalization, or functional block. And that inhibits acetylcholine from binding its receptor. It inhibits the downstream signaling of the neuromuscular junction, and therefore impacts things like breathing and swallowing that can be incredibly important, obviously, for quality of life and living in general. And what is really critical here is that one of these mechanisms is FC mediated. So again, our method is the only method where you get FC level resolution. And there's a lot of evidence in the literature that there are particular FC alleles that may be associated with enhanced complement activity. So our translatable potential here is that if we can identify an allele level risk potential, so maybe there are alleles that are associated with more complement fixation that suggest maybe there's more risk of that to become an autoreactive allele, maybe with enough functional evidence and enough support, those individuals that carry that germline can either be targeted for more specific healthcare or maybe treated with those complement inhibitors. A complement inhibitor isn't going to help people who have the sort of internalization mode of MG, right? But a complement inhibitor will specifically benefit those people that have a complement mediated MG. So we started looking at this. This was a study that was funded by the MGNet Rare Disease Network. So thank you to them for their funding. We got a small cohort of AChR MG individuals, 48, and compared them to the antibody profiles and the germline profiles in a healthy donor cohort that was age, sex, and ancestry matched. This is just an example of a tile plot of some of the germline genotypes that we saw. It's a big mishmash. The take-home message is in the legend. If you look at the legend and the colors that correspond with anything that was named FL, that means that those are novel alleles that were not seen in the database at the time. So again, every time we sequence someone at the DNA level, we identify new alleles, even new genes. And this draws a lot of questions also about the quality of our reference database, not within the scope of my talk today, but something that I would love to talk to any of you about. It's really important. We also see the presence of more than 2 alleles per individual in some cases. This would be unusual. Initially, we thought this was an artifact, right? We all have a maternal and a paternal allele. But what we did find when we saw this was that there are specific gene cassettes, quite a few actually enriched in the IgG4 region of the IgH constant region, where there are gene duplications happening as well, leading to the expression of additional alleles in the repertoire. Now, whether that contributes to pathogenesis, if these happen to be more autoreactive, that's still an open and tantalizing question. If you look overall at sort of the number of novel alleles that we found, the majority of them were novel, not curated in IMGT, which is the immunogenomics database that we all tend to use as a reference. In fact, between 60 and 80% of the alleles we identified were novel compared to what was known in the database. Again, critically pointing out that more resources are needed, and it's something we're actively working on. So with the last few minutes, I just want to tell you sort of where our future directions are going. We're using this constant region data to annotate those constant regions. There's a lot of literature out there in the antibody engineering field in particular that have made specific mutations in the constant region to try to enhance activity, and then observational data like the studies I'm highlighting here that have seen that there are changes in the constant region that can impact function. So specifically in these 2 studies that are from 1990 and 2021, so 30 years apart, show that deletions in the hinges of the IgG3, so these are small stretches of amino acids that allow the head of the antibody really to twist and turn and open up for antigen binding. When you delete that and you restrict the movement of the head of that antibody, you actually enhance complement fixation, binding to C1q, and complement activity. And since we know that our disease is complement mediated, we wanted to specifically look at this deletion in our repertoires. And so we went back to our cohort and to healthy cohorts as well and tried to map the relative frequency of the M allele, the major allele with the 4 hinge regions, and then the short allele that has the 3 hinge regions. The first thing we did was just to understand, does this differ across populations? And we looked in a healthy cohort. This paper actually was just posted in bioRxiv a few weeks ago. We did 105 individuals, just their constant region genes, so 210 haplotypes, and we saw a very different variation. Here, those blue bars that are a small fraction of the populations, those are people who have 2, 3 alleles, 2 short alleles. The pink is when you're heterozygous, 1 long, 1 short, and the green are the long alleles. But you do see, again, across those major global populations that these differ per population, which could, possibly explain different susceptibilities in populations to autoimmunity, which is something exciting to follow up on. And then we looked in our MG cohort in particular and saw the enrichment of these structural variants in this cohort. So anything that's not that blue color, which represents 2 4s or 2 major alleles, represents the carriage of at least 1 allele that has that structural variant. And actually, in a few cases, we actually see some of these duplications that show the carriage of more than 2 alleles, showing that IG3 has duplicated in this locus, and that maybe that additional third allele circulating is also contributing. So lots to follow up on, lots to explore, very suggestive, but very exciting. And that this is what it looks like. So if you schematize these duplications, right, the references on the bottom, this is the order of those constant region genes. And we've seen structural variants where one or more than one of those constant region exons are duplicated at the DNA level. So after all of this data, we've been collecting this, we've been working on these assays for over 10 years now, as I mentioned. My collaborators and I really started to see the potential of these, and we wanted to be able to offer them to everyone else. We think that when you think about effectiveness of drugs, a lot of the focus is on the formulation. Did we add the right adjuvant? Did we target the right immunogen? And very little attention is paid to how different host immune systems may be influencing that activity. So we want to use this platform to introduce our company, Clareo Biosciences. We've developed first-in-field long-read-based assays for both antibody and T-cell receptor profiling, all 11 loci, so light chains, heavy chains of the antibodies, as well as TCR, alpha, beta, gamma, that corresponds with those germline assays at the DNA level. And together, if you use them in what we're calling the Clarity package, you generate a personalized genome reference that then you can map your repertoires to have the most absolutely, the highest accuracy and comprehensive coverage of the immune repertoire. And we really believe that these data can enable the search for biomarkers, clinical correlates of protection, as well as get at the root of what mechanisms of disease and disease pathogenesis are. And together, we think this data allows people to do sort of three major things, right? The first thing is to discover, are there immune loci? Are there immune variants that are associated with my phenotype of interest? If you are studying a disease where the modulation of the immune system is part of the pathogenesis or part of the therapeutic effectiveness, then I would bet money that there's an immune gene that influences it. Once you discover that gene, you can use our tools to screen inclusion-exclusion criteria for clinical trials, screen individuals retrospectively to try to post hoc explain phenotypes and results. And we are hopefully aiming to develop AI/ML algorithms to use this to develop predictive biomarker screens. And then, of course, our moonshot would be able to use these data to inform pharma and other drug development companies on how to design drugs that have the most breadth across populations with the least amount of side effects. So let me tell you what our vision is about sort of how we can use our tools to enhance therapeutic effectiveness for all. So the first, again, is in that preclinical candidate design and selection process. So if a company has 5 therapeutic interventions, 5 drugs that they're developing, they've all gone through sort of that preclinical evaluation, we can come in, we can screen those samples that have been used for preclinical evaluation, and we can say, okay, candidate number 3, that has the most activity across the widest populations. Or candidate 5, it has the highest risk of eliciting a response corresponding with those genes that are responsible for immune-related adverse events. So let's move 1 or 2 of these forward, or maybe back burner one of them. From a pharmaceutical perspective, this should enhance time to market and decrease cost of development. Once you are in the clinic, either at the trial stage or at the actual clinical treatment stage, we would love to be using our tools as a companion diagnostic. If we know that we can predict immunotherapy response, vaccine response, or the chance of side effects, we should be using our tests to be able to bin people appropriately for clinical care or include them/exclude them from clinical trials that may pose a risk to their health. To that point, there have been many clinical trials that have been shut down early due to excessive amounts of immune-related adverse events during that trial period. So maybe we can go back to those trials and examine retrospective samples, identify what those immune signatures were with that immune-related adverse event, and maybe rescue some of them, right? Maybe find certain populations where those drugs are really effective in, where they won't have immune-related adverse events. So maybe some of the money and time that's already been invested in development can be saved or salvaged through this type of targeted evaluation with retrospective samples. So with that, I'd like to sort of conclude with some final thoughts. I know that my name is everywhere on this announcement, so please reach out. I would love to talk to you all about your thoughts, about any interest in working with us about precision immunogenomics. So first, I really want to emphasize, and this is why I'm so excited to be presenting together with PacBio, is that highly accurate long reads were absolutely essential for resolving these loci. We could not have done it without HiFi. Like I said, we've been working on this for 10 years, but it really only came together post 2019 when HiFi was made available. So thank you to PacBio for bringing our lifetime work to fruition. I think another thing I really want to drill home, and I know I said it a lot in the data, immunogenomic variation matters, right, for response, but also data representation matters. The different distributions of variants across populations is going to be critically important for us to understand why certain peoples are susceptible or resistant to certain diseases, and we should be considering that in the drug design process. Understanding the extent and impact of this variation in immune responses will be critical for precision genomic medicine. So many of the drugs that are available on the market today somehow manipulate your immune response to get their therapeutic effect. And that underlying foundation of what your immunogenomic DNA looks like will impact that. So we really need to make sure we're considering that both in the design and the treatment with these drugs. And that this impacts both T cells and antibodies, including responses to immunotherapy, with data I didn't have time to show you today, several different types of vaccines. And so, this just isn't a one-off. There's a lot of potential here. There's a lot to explore here. So with that, I want to thank everyone for their attention. Definitely acknowledge my co-founders at Clareo. I'm putting a QR code up here that you could, screenshot and come find us and talk to us later, as well as my email. Feel free to reach out. And a lot of this was based on the foundational work we have done at the University of Louisville. So I also want to thank both our Louisville teams, our collaborators at Yale, and the funding for the studies I presented today. So with that, I would be delighted to answer any questions.
Thanks, Melissa. Everyone, please use the questions box to ask your questions. We already have some questions coming in. I will start with this question. I was wondering how the technology and the concepts discussed here have been applied to analyze the effectiveness of oncolytic immunotherapy agents such as TVEC.
Well, first off, I want to really thank everyone for your thumbs up and your applause emojis. It's great to encounter an audience that appreciates the work that we've done. So we actually haven't had the opportunity yet to work on the oncolytic therapies, and we'd be super excited to do so. I think for me personally, trained as a viral immunologist, those are really exciting for me. They sort of cross both the oncology space and the virologic space that I'm historically working in. So the person that asked that question, please reach out. We'd love to chat with you about it.
Awesome. The next question is, thank you for the great talk. How much impact does long-read sequencing have on air coverage, the adaptive immune repertoire coverage, and overall cost compared to short reads?
No, I think this is a really important question. And obviously, as much as we'd all like to design the optimal study for rigor and reproducibility, those budgets are always a constraining factor. I think, this is somewhere where I really have to give a hat to PacBio, because the development of that Kinnex assay really changed the landscape in terms of our throughput and cost point. So the first paper that we released about FLAIR-seq in 2023 in JI was the single transcript per well, and it was about 2 times the cost of doing the same type of profiling with Illumina. But now with Kinnex, when we can concatenate 8 transcripts per molecule, it actually ends up being cheaper than short reads. And then you get the more information of the constant region on top of that. So comparable coverage, if comparable to more coverage and comparable to less cost. So there really isn't any practical hindrance, minus the fact that you just are going to discover a lot of new stuff. And that's what we're here to help with. Something I didn't mention in the talk, but I do want to highlight here is that, our group, William, Corey, and I are, experts. We're part of the AIR community. We really want to help get the news of this data out there. And so we're happy to also work on, computational only projects for people who are really interested in trying this out. We're happy to do those types of comparison data sets as well.
That actually leads to a nice question is, how might researchers access your technology? You've mentioned ClareoBio, and you've published both the iGenotyper and the FLAIR-seq, which are now called Profire and Genotype Air, as part of ClareoBio's offering.
So that's exactly right. I think, we want to make it as accessible as possible, which is why Clareo came to be. Because a lot of people don't want to stand up new pipelines in their lab when they know that there's someone out there that can run them quickly. But those pipelines are both published and open for academic use. So if you want to stand up the assays, feel free. We encourage anyone to really dive into this area. We think it's really important, and no matter how you get your data, we're excited for you to explore the area. If you want to do just a one-off project or a pilot just to evaluate what more it can bring, then that's what we're here for at Clareo to do.
Awesome. We have another question. Have you looked at assessing clonal abundance using this method for diagnostic characterization of lymphoid neoplasms? Or how about akin to what Adaptive is doing using this for looking at MRD?
Yeah, no, that is definitely on our roadmap. I think, we're in the process of, we're relatively new. I think about 6 months in, we just opened our lab in February. What's actually been the biggest challenge, which is one of those, what's the word I'm looking for? It's a privilege, but also a challenge, is focusing, right? And finding sort of the lowest hanging fruit that we can really make the biggest impact in straight away, which is sort of this service, getting the news out there and spreading the word, which is why we were so excited about this opportunity. The MRD is on our roadmap. We think that the additional variation that we see both in the genomic and repertoire profiling with the sort of just additional content that we screen will give a higher sensitivity to assays such as that. And that is definitely the direction we're heading.
Thank you. I will combine the next two questions as they are related to each other. What are the key computational challenges in analyzing HiFi for immunogenomics, especially in assembling and annotating the immune repertoire?
Right. So there's sort of two answers to that. So the first is really, the dearth of in the reference databases. I mean, IMGT has been an incredible tool. They put in an incredible amount of work. But as I mentioned in the webinar, every time we sequence a person of whatever ancestral background, we discover new things. And I get, very concerned about comparing to an incomplete database and sort of miscalling genetic variation as somatic hypermutation when you're doing these types of inference methods, when you're using RNA sequencing and inferring backwards, which is why we're really excited about offering that paired personalized reference for those really important cases where you want to really know, is that just crazy extensive hypermutation, or is there really new genetic variation here that we need to reflect? So the biggest computational challenges for us is building up that reference database. And, at the risk of plugging Clareo too much, that reference database will be a product in itself. So we're going to produce a database that is queryable for the community to use as well. And actually, maybe not so much with Clareo, but partnering with other databases like BDJBase and OgreDB to get that data out there so that everyone can have access to it. And that's critically important. And on the academic side, we're funded to do that kind of population genetics work. What was the second part of it? Oh, and then the repertoire. So I think the key thing here, right, is that we're not assembling. And I think that's another feature that a lot of people who haven't used long reads don't realize, is that you do get that full molecule as a single molecule. There's no assembly required. There's no reconstruction. And there's no reference-based sort of, it's all de novo analysis. Yeah. You don't have to use a reference-based approach. And so actually the FLAIR data works and goes right into, say, the incantation pipeline available through the Yale group. And the details of how to use that FLAIR data with the incantation pipeline is in that JI 2023 paper. So we detail there how we do that informatics sort of step by step. It goes in right after the assembly step. So in that pipeline was built for short reads. They assemble the paired end reads. We just bring in those HiFi reads underneath that step, and then the annotation goes from there with all the incantation tools. So it's completely compatible with the sort of Presto, Chango, Alakazam packages available through incantation. The assembly on the DNA side can be challenging, but mainly because of those reference-based issues with the annotation. But assembly, we're using the same tools that anyone uses for genome assembly, HiFi ASM, and things like that.
I see. Well, that actually answers the second question. Someone had asked where are the analysis tools for these, yeah. But then you've just explained that for FLAIR-seq, people could look at the paper and it sounds like incantation would straight up work. And then for the genome assembly, a standard PacBio genome assembly tool such as HiFi ASM would work. And I assume that if we go and read the papers, they would find that as well.
Yeah, absolutely. The iGenotyper paper from 2020 that first described that capture genotyping method's a bit old. I mean, at this point, it's 5 years old, so the tools have definitely advanced since then. So, feel free to reach out. We can help with as much sort of guidance as we can, but we do use HiFi ASM as sort of our backbone for that. I think the key is really figuring out how to annotate effectively, which is, why, again, we built what we built at Clareo, because we want to make sure that not only is the most accurate data out there, but it's in a state that the community can use as well. We're very dedicated to making these resources available to the community, both on the academic and the company side.
Awesome. Well, thank you so much, Melissa, for joining us. And thanks to all
It's been such a pleasure.
join the webinar and still staying on. I hope to see you again. Bye.
No, thank you so much. Bye.